Okay, I'm recording this session now. अस्सलाम वालेकुम, बिस्मिल्लाहिर्रहमानिर्रहीम। मेरा नाम डॉक्टर इमजाद हुसैन है और मैं आज का सेशन जो है, आज का टॉपिक है एवोल्यूशन ऑफ viruses, especially SARS corona viruses, and we'll be looking at it from host virus perspective. And in host virus perspective, I'm hearing some noise. Probably somebody's microphone is on. Please mute yourself. So we're talking about host virus perspective. Okay, so this is next screen. I just wanted to show my biosketch here. That's on Google site also. I'm doing a master's in health professional education. I'll try to be brief about my biosketch. I'll just run it down. These are my credentials. I was born and raised in Jhang, Pakistan. I did MBBS from Mr. Medical College in 1999. I was ECFMG certified. I did USMLEs and got an ECFMG certificate in 2004. I have been a clinical research fellow at NYU MC in Aging and Dementia Research Center. And I also worked as an assistant research scientist in Translational Disorder Research Center. That was to do with, mostly to do with prion disease. And I passed my step 3 in 2008. Can somebody mute himself? It's creating noise. I don't know. Okay, let me mute someone. Let me mute all. Please mute yourself. Okay, I will manually mute it. Sorry, there was some noise interference. And so I did step 3 in 2008, and I had a postgraduate certificate in family medicine from UHS. And I also did a certificate in neurosciences from Duke University. And last year I completed a mini fellowship in movement disorder from University of Nebraska Medical Center. Currently I'm doing a master's in health professional education. So these are my credentials. Let me admit another participant, sorry. I'm being continuously interrupted. These are my certifications. So Charles Darwin said something beautiful about evolution. In the long history of humankind, those who learn to collaborate and improvise most effectively have prevailed. So probably that's the main quality in human being, and that's why we are prevailing. I will again ask people to mute themselves if they can, because it's creating interference. Kindly mute yourself if you can.
सर ये एक आईफोन के नाम पे आप खुद उसको म्यूट कर दें।
Okay. Okay, I'm trying. I think it's muted. Okay, so we're good to go. So I hope you can hear me. So this is my YouTube channel link. I've edited various videos on that. So I'll go beyond that. So let me just let me admit somebody. So evolution is the process by which different kind of living organisms are believed to have developed from earlier form during the history of Earth. So this is what evolution is, and it is a process of change. Evolution is actually the net directional change for any cumulative change in the characteristic of organism and population over many generations. So when we say evolution, just like in terms of this virus, when we say this virus is evolving, the change has to be in terms of characteristic. The change has to be phenotypic. It has to translate into change characteristics of virus. So we'll go to next slide. I have problems switching between slides. So learning objective here are, uh, I'll leave you with, as I said earlier, I'll leave you with more questions than answers. So our learning objective are viruses and our relationship to them, origin of viruses, SARS coronavirus 2, viruses as a morphological entity. I will discuss the SARS coronavirus structural molecular genetics and evolutionary arm race between virus and humans, and the mutational potential and the quasi species, and the post-COVID world and open science. So this is a rough sketch of virus. You can see how large animal cells are. Animal cells are 10 to 30 micrometer, and viruses, even the largest of them, are, you can say, like 600 nanometer. And HIV, for example, it's only 120 nanometer. Rhinovirus is like 30 nanometer. This is one of the, I would say, one of the giant viruses. It was recovered from permafrost. Permafrost is something that we get from glaciers. So it's believed it has been here for like eons or centuries. That's another big virus. It's called virus, and it's about the size of, we can say, 500 to 800 nanometer. And when we compare it to the SARS coronavirus is hardly 135 nanometer. So it's a huge virus. That's another virus that is being virophage. So virus actually that phages a virus that actually thrives in another virus is a virophage. Sputnik, that's 40 nanometer. And that's a kind of overview of size of viruses. You can see how big is virus and how small is poliovirus here. And these are various shape of viruses. You can look at the Ebola virus and other viruses that have, uh, like you can see, they have spikes on them. MERS, SARS, and other viruses you can see here. There's another uh, uh, overview of viruses. It's showing size of viruses, and not just the size of viruses, it's showing uh, the years when uh, those viruses were causing infection, when those viruses were causing pandemics. And for example, the Russian flu that killed 1 million people, you can see Hong Kong flu 1 million people, and that was in between 1950 to 1975. And these are, this screen is showing DNA and RNA type of viruses. So I would also like you to see another important thing here, that is like RNA viruses have two polarities. One is a positive polarity, another is a negative polarity. Positive sense RNA viruses and negative sense RNA viruses. And rest are DNA viruses. I won't go into detail. So I'll briefly touch the paleovirology. What is paleovirology? The study of track record of viruses origin based upon genomics. So basically that's how they study virus. They study genomics. And then molecular fossils. Molecular fossils are part of virus genome incorporated into host genome and then passed on to another generations. Like, for example, viruses do live in our genomes. And for example, if you look at the HIV virus has an enzyme that's called integrase. That's how it integrates its genetic message into our genetic message. It infects every cell and it stays there. Brachyviruses have shown that viruses were there for more than 310 million years ago. So what is phylogenetic? We'll talk more about phylogenetics. These are various forms of life. You can see the bacteria, viruses, eukaryotes, archaea are something, and that's another screen. It's showing how about the tree of life, how the uh, life evolved on Earth, and where do viruses stand. Viruses basically are regarded nowhere uh, in tree of life. They are regarded more like, I would say, just like a liana tree. Liana tree is a, जैसा कि आप देखें तो ये आकाश ट्री जिस तरह से होता है कि it's entangled, it's intertwined upon all the trees, box, and that's how it. it basically thrives upon other trees, and it's attached to kind of other trees. So viruses are like that. So let me go to next screen. I'm not able to switch screen. Something, yes. That's the basic difference between eukaryote and prokaryotes. It's based upon the nuclear material. If it's nuclear material is loosened, these are prokaryotes. And animal cells, they do come in, basically they have DNA and RNA that's enveloped. And if they have loose material, then they are like eukaryotic cells. So we'll talk about origins, last universal common ancestors. Uh, that's what we believe from evolution, that we have some universal common cell as an ancestor. And here it's showing GAs. GAs are like uh, giga anoms, thousands of centuries, I believe. That's what it means. So we see three main classes: archaea, eukarya, and bacteria here. And this is something about the time period in history: ages, epochs, periods, eras, and eons. So life on Earth was probably like 45 billion years ago. That's what we believe that it started. And so who came first, viruses or us? It's an age-old debate going on for centuries. There are various hypotheses about origin of viruses. Some people say, some scientists say, like viruses, they came first, and they were self-replicating units, simplest form of life, and basically they were relics of time. And then there is another hypothesis, it's called escape hypothesis. The escape hypothesis says that viruses weren't the first to inhabit the chain of life. They basically, they were like an excrement of genetic material from other cells. Uh, while they were replicating, their genetic material slipped off the assembly line. And I already told you viruses are like liana on trees of life. And here you can see the lianas. If I can show you the branching pattern, and then there is something uh, like in the form of waves that's intertwined upon these archaea, eukarya, bacteria, and it's like a hexagonal structure in there. It's called the horizontal gene transfer, HGT. So Luca means the last universal common ancestor, and then Lika is last eukaryotic common ancestor. That's how organism evolved. So I'll leave this detail. So viruses are within us. 80% of our DNA, uh, sorry, not 80%, about 8% of our DNA, human DNA, it's made up of viruses. So viruses are like ghosts that are living in our cell, and we don't really know about them. That's phylogenetic tree of viruses. Viruses, basically, they are not included in tree of life, so they are classified separately because viruses, they do not have a common origin, and their even genetic material is from, it said, their genetic material is polyphyletic. It's from various origins. It does not compare to a single gene even. So viruses are alive or dead. It's a long debate. Some people say like they are alive because they have certain characteristic, but most of the common thing that are attributes of life, viruses, they do not have them. So we'll talk about structure and biology of molecular genetics and biology of SARS coronaviruses. Coronavirus is the family, and there are 7 coronaviruses till date. And 4 of them we know they cause common cold, and 3 of them you know that the latest one is novel coronavirus or SARS coronavirus 2, and the previous 2 were SARS coronavirus classical and MERS coronavirus. And they were both like deadly viruses. They have high death rates. So talking about structure, we can see the spike structure because of these spike protein. We can see that's how the virus derives its name. We call it uh, coronavirus because of the coronum, the spikes on them. Spike protein is very important in terms of uh, virus transmissibility and how it gets into human beings. Spike protein, it basically binds to ACE2 receptor, and that's how it gets entry into human cell. It has other components like S1 and S2, and it has various regions. So it has other protein too. It has nucleocapsid, membrane protein. Nucleocapsid protein is basically bound to the nuclear material as well as the membrane. And then M protein keeps basically the structure of virus intact, and envelope protein. So it's a, I already said it's a positive sense RNA. Positive sense RNA means that it directly translates into protein. It directly latches onto ribosomes. And it's a 30 kilo base pair means it has 30,000, its genetic material is 30,000 base pair. And its size is 125 nanometer. It has a lipid bilayer that is basically prone to chemical damage by even soap. It can be damaged with the help of soap. And spike protein, we already discussed, that's how virus gains entry through into the human cell through the receptor binding domain by attaching to the ACE receptors, ACE2 receptors. It's very important. So, so far, I think we have like 10 or 15 minutes more. Okay, let me switch to the next slide. So this is the receptor of, this is the structure of spike protein, and spike protein has a receptor binding domain. Uh, this receptor binding domain is basically, it's very important in terms of viral entry, and also it's important in terms of vaccine development, because even monoclonal antibodies are being designed to cover and block this receptor binding domain so that virus cannot get entry into human cells. So it's very important in terms of drug development even. So here I'm showing you genome, that is I said 30,000 base pair, and the proteins, basically it has 29 protein. ORF is the open reading frame. I won't go much into detail of it at the moment. I'm short on time, I believe. So origin of coronaviruses, we already know there are 7 coronaviruses. We said, and most of them you can see their reservoir is among the bats. So alpha and beta coronaviruses, basically, they are the main viruses uh, that cause disease in human beings. So there is an intermediate host before the virus, before the SARS and MERS and other coronaviruses, they cause disease in human beings before they are able to jump into human upon human species. So for bats, we know there are more than 400 coronaviruses. So bats is a big reservoir. Bats do cause other diseases like Nipah, SARS, Ebola, Hendra, Marburg. So bats are like a, I don't know how bats harbor so many viruses without causing any deadly disease to them. That's $1 million question. So this is a spillover effect. I'm showing you spillover effect, how uh, viruses are from zoonotic viruses are able to jump to human beings, but they require an intermediate host for the spillover. So spillover means virus to be able to jump from one species to another. So to be able to jump from one species to another, it has to have mutation and some sort of, I would say, receptor change. Most of the coronaviruses, they do cause disease in humans through ACE2 receptors. Same for the MERS and SARS. So again, intermediate host in this case is not, we're not really sure whether it was pangolin or snake, but for reservoir host, we know that was the bats, horseshoe bats. So this is basically life cycle of virus. It's shown in 8 circle, how virus gains entry, then it attaches to ribosomes, and it shuts down basically host genome, and in a way, it kind of hijacks host cell to basically manufacture its protein, and that's how we see, like here we see the subgenomic RNA and genomic RNA production, and then the assembly, and lastly, uh, when a complete virus particle is created, it's excreted from the cell through exocytosis. So that's again various segments of gene and how they translate into various enzymes. These are very important enzymes in terms of drug development, and especially for repurposed drugs, how drugs interact with virus. It has endonuclease, it has exonuclease. Somebody raised hand, but I think I will be able to uh, address that issue. Let me just finish up with the presentation. I do not have a host today. From next time, we will have a separate host and a moderator. So it has like 14 open reading frame and 27 segments, UTR or untranscribable region. So what's important here is non-structural protein and structural protein. Non-structural protein have the RNA-dependent RNA polymerase, and structural proteins have structural protein give a lot of characteristic to viruses, and that's why this virus is so evasive because of non-structural proteins. And I'll just run this down. So this is another uh, uh, non-structural protein complex. It's a NSP12 polymerase, and the rest of the NSP7, 8, 12 that makes up an enzyme, hollow enzyme that initiate de novo RNA synthesis. And that's another mechanism virus has for mismatch repair. Uh, Probably I might mention this virus has a proofreading mechanism. That means that if this virus is copying its genetic DNA and somehow there is an error during copying, it has a potential to kind of correct it, auto-correct it. But still mutations keep on happening despite this mechanism. So NSP1 is something, the non-structural protein that actually restricts human or host cell gene expression that in a way blocks our immune response. Interferon, we know a lot about interferon. These viruses and the MERS coronaviruses, they basically induce very little interferon. Maybe that's one of the reason, that's one of the success story of their pathogenesis. That's why they are so sneaky in causing disease. And they just happen to go inside human cells, and when they are detected, they already have made a lot of progress. So it's again delayed interferon IF1 signaling, and there is dysregulated inflammatory monocyte macrophagy response. So that's the main reason for cytotoxicity. And dysregulated immune response that leads to exuberant innate response. So there is a lot of damage caused to human cells because of this dysregulated response. These cytokines, chemokines, they start damaging their own cells. So that's why we see the CRS or cytokine release syndrome. So that's this slide is showing like host factors are also responsible. For example, SARS coronaviruses, they do get into human beings through ACE receptors. So different people have some pleomorphism in their ACE receptors. So that's why maybe different people are getting disease differently. Elderly people have kind of severe disease, and people with hypertension, COPD, and diabetes, cardiovascular disease, and they do have severe complications like septic shock, and they do have coagulation, multi-system organ failure. So this is again CRS, cytokine release syndrome, recipe for disaster. So in a nutshell, let me just read this one. This is a very good slide. So what all this means for our immune response to SARS coronaviruses. So what we know so far is that these viruses, they do generate reactive immunity, and probably most of this immunity or antibody will disappear within months. Apparently, humans respond to SARS coronavirus very differently, very heterogeneously, and some people, they do not even successfully launch immune response. And in secondary immune reaction to SARS coronavirus, people, they still might be infection. Like for example, they just recovered from infection and they could again have infection from the SARS coronavirus. So then there is an intentional natural infection. Intentional natural infection uh, is like people are getting infected just for the sake of getting herd immunity. That's not a really successful experiment so far. It's very dangerous, and virus do have a mechanism of tricking immune system. That's what we said, the immune evasion. So a vaccine can be tailored to stimulate an efficient immune reaction and avoid detrimental ones. but we do not know how effective our vaccine would be. That's a breather. I just added a picture of me from last year. I was getting a movement disorder certificate from Dr. Danish Pati. I learned a lot from him. So we'll talk about mutations briefly. That was the main topic. So, like all RNA viruses, they do have mutational potential, and so the SARS coronavirus is also gathering a lot of mutations. So what we see here is virus is like it's infecting different hosts and it's getting errors or it's getting change in its genetic material, and we are calling it mutations. So technically virus is mutating, but it's not causing consequential mutations. Consequential mutation means something that have an effect on its transmissibility, its infectivity, its disease pattern. So there are probably more than 200 sites across the SARS coronavirus genome, and there are 7,500 of SARS coronavirus 2 genomes. So far, there is a clearinghouse or there is a hub for these genetic mutations. I'll show you. It's coming. And TMRCA analysis, that's like a time to the most recent common ancestor analysis. We'll talk about it later. So this is like a genetic map. You can see, for example, X. Oh, sorry, I skipped it. So for example, this person has an X virus, and X virus is infecting two people, and red and the blue one are its genome, so they are kind of same genetic material. And then they sort of mutate during copying and they change into A and B, and they are shown by different colors. So that means they have mutated, they have significant mutation. And the other one again, the common red one, that mutates into uh, D and E, and then another mutation happens from D, and this one is called the C. So that's how we make genetic map, and we are learning about spread and different strains of viruses. GSAD is the clearinghouse, or it's the basically it's a let me say it's a resource for all strains of viruses. People are sending their blood sample. Institutions are sending their blood sample for whole sequence genome analysis. And this site or this hub is a German site. GSAD is doing the phylogenetic analysis on them. It's doing even short read analysis and recombination analysis. So mostly they are seeing is point mutation, and you are seeing various colors and a kind of pattern, like something is derived from one, uh, like blue thing is derived from red, and that blue genome is kind of converted into some greenish and yellowish dots, and then dots are kind of larger in size. For example, red dot is really large in size than the blue one. So these are various strains of viruses. We may not really call them they are truly mutated or they have the potential to cause different disease, but there is some difference. So this is the genetic epidemiology of novel coronaviruses and global subsampling shown from the GSAT data from they are doing it from 2019, 3rd December to up till March, and probably I think they are still doing it. Mine is a kind of I did an old version. So virus surely is mutating, and mutations are in various regions, and this one shows the diversity of mutations. Open reading frame 1B, open reading frame 1A, and then the S even, and we know that the spike protein is gathering mutation. So that could be causing a consequential mutation. If somehow spike protein gathers a mutation that makes it impossible for the virus to attach and infect human cell, that is called like a detrimental mutation, and virus is going to lose its infectivity. We're not really sure up to what extent these mutations are happening. That's again the same thing I told you. It's just like flying of aeroplanes from or the air traffic from across the continents. And in a way, it's comparable to that. And then these are quasi species or strains from A, B, and C. I'm saying it quasi species, and most of the scientists are saying them to be quasi species or strains because they are not truly uh, like different viruses. They just have, for example, the type A, that was the, let me say, that was the first one that was closest to SARS coronavirus in bats and pangolin, and it was it was the root of outbreak basically. This virus was seen in Wuhan in uh, earlier explosion, earlier disease, and then it kind of escaped somehow. And it gave rise to the B form, let me call it daughter for that, daughter from the A. So it has variation, and it has a lot of variation from the A variant, and it was derived from basically it has two mutations, not a lot of mutation, it has two mutations, and this was kind of a bit less, causing less deadlier disease than the other one. Then type C is seen mostly, uh, it's seen in Europe. That strain is seen in Europe, and we can call it, it's like another daughter from the type B. So these are causing species. This one shows a geographical map, and what it's showing is color coding of various strains of viruses from Oceania. It's blue, Asia is light blue, Europe is yellow, and North America, it's like an orange, and red is for South America. So if we see uh, after 5th of February and up to 26th of February, there is kind of an explosion of mutations. It's like virus is kind of radically changing. So coming back to the topic, what are clades and superclades? We're calling them like strains. So far there are like 10 strains according to the GSAID data, and from the nextstrain.org. Basically 10 basics, and among 10 basics we can like then clump them into two major. So we can say like A1, A2, A2A, A3, A6, A7, it's like a superclad A, and then the B1, B2, B4, we're putting it into a superclad B. We can say like one is an Asian version, another is a European version. So these clades are based upon the phylogenetic analysis. They're just not randomly saying that uh, they are causing mutation in European and East Asian countries. They are behaving differently. This is the article. I was just going through spike mutations. Pipeline reveal emergence of a more transmissible form of SARS coronavirus. So remember, I was saying spike mutation could cause a virus even to become less deadly or incapable of causing disease. So on the other hand, spike mutation could lead to more transmissibility. I'll just skip the details. That's one of the beautiful songs from the Gora Halova, Sheher Khali, Jada Khali, Kucha Khali, Khana Khali, Persian. Probably she was a Persian singer. And it feels like this song was all about the social isolation and this post-COVID world. So medieval time, we see what was happening there, quarantine, and what's happening now, quarantine. So what's important is breaking up the barriers or breaking up the silos is what's more important, like we are doing today's. We are breaking the barriers. We are kind of coordinating with each other, and same thing in Europe and American countries. What's the major issue is that institution, they do have a patent policies. Even scientists, they do have a patent policy. If someone is discovering a substance or drug or something, that will be patented to them. So in a way that makes people kind of less collaborative. They do not collaborate. So these are silos. Silos are like, you can say, they have they are like storehouses for grain and other things. These scientific silos, they need to be collaborated. Different institutions should collaborate. So this is one example of collaborating. They are enabling rapid drug repurposing. And this is one of the University of California, San Francisco department that is collaborating with like 130 labs, and it's collaborating across the globe with one institute, Pasteur, and another, like one in Paris and one in Germany. So this is what collaboration leads to. So there are now more than 2,000 FDA approved drugs for SARS coronavirus. that are kind of in pipeline. They are, I think they all of them are not really approved. They have different levels of approval, and 10,000 clinical stage drugs, and then preclinical drug candidates are much more than that. I think this map is like how various receptors and chemical substances they interact with each other and with all these receptors. So it's basically how technology helps us. We see here 69 existing targeting SARS-CoV-2 2, human host factor, and approved one. We can see chemostat, linezolid, ruxolitinib, verapamil, tiagycilline, valproic acid, and then the one in yellow, they are in clinical trial. Most of them, they do not have a name. They just carry a kind of chemical number plate or abbreviation for that. And then there are many preclinical substances we see in the pink color here. So what happened in the Spanish flu shouldn't be called Spanish though. That's how the word remembers it. Uh, it was a kind of global event. It has nothing to do with Spain. It has like three, I would say, three spikes, or there were three waves. And this is, I was just listening to a lecture of Laurie Garrett. She's a fabulous writer. And she wrote so many books, and she was warning like at least 10 years before this epidemic came, coming black. And this is me again. I'm also an editor at MedShare. So that's it from my side. Thank you very much. So we'll start with question answer session if we have some time. So we all can unmute ourselves. Okay,
That
please.
was a very good presentation.
Thank you very much. I just tried.
जी काफी इंफॉर्मेटिव और बड़ी अच्छी प्रेजेंटेशन थी अमजाद भैया आपकी.
Thank you. So do we have something to discuss upon? I know it's a dense topic. I may not cover all the details. It's from many disciplines, like it's from biology, it's from genomics, it's I would say even from history,
अच्छा आपने ना
virology.
डिफरेंट टाइप्स बताई थी इसकी स्ट्रेन्स वगैरह ना ए बी सी डी कि एशिया में मुख्तलिफ थी, यूरोप में मुख्तलिफ थे. क्या इसका कोई इन्फ्लुएंस है पेशेंट के ऊपर चेन का कि उसके कोई साइन एंड सिम्टम्स बदलते हों या सेम इफेक्ट है, सिर्फ नाम बदला हुआ है अकॉर्डिंग टू द ग्राफिक, कुछ ऐसा?
Dr. Farhan, something about strains, ऐसा है कि we are saying them quasi species. We are not really saying that are altogether different virus. Like so they do have some changes that that's causing different disease pattern. For example, we see less severe disease in some countries and we see more severe disease in other countries. But there are more confounding factors. For example, different countries have different healthcare setups. Different people have different propensity for the disease, and there are not many. I think host factors are far more important than virus factors. So far, there is no scientific evidence that virus is weakened somewhere. These are merely strains. So hope that addresses your concern.
जी जी बिल्कुल.
And let me add
चलिए
just
माशाल्लाह सर बहुत अच्छा
जी
सर बहुत अच्छा माशाल्लाह रहा. मैं तो आई एम सॉरी दरमियान में मुझे ना आईसीयू वाले भी डिस्टर्ब करते रहे तो मैं कुछ आपकी बातें सुन भी नहीं सका. दरमियान में कॉलें भी मुझे आई, लेकिन ये है कि जो भी मैंने सुना है माशाल्लाह बहुत अच्छा रहा. बस वो हमारी कोशिश ये है कि ये स्टार्ट एक हुआ है ना, हम इसको कंटिन्यू रखें. इंशाल्लाह मिल के चलेंगे तो बहुत अच्छा रहेगा.
जी बिल्कुल इंशाल्लाह we should keep doing it and each of us can update each other. Dr. Lukman can update me far better then myself, we can talk about dermatology. Dr. Akmal can update about other aspects of the disease. And so that's how we collaborate. I think that's how we should do it. It's not like a lecture.
सर एक
I'm
और
not
क्वेश्चन
a.
था,
जी sir. Go on.
आपने बताया कि ये म्यूटेट कर रहे हैं सर, वायरसेस म्यूटेट कर रहे हैं डिफरेंट एरियाज में, डिफरेंट तरीके से म्यूटेट कर रहे हैं, तो इसमें हम कैसे जान सकते हैं कि ये फ्रेंडली मैनर में मतलब म्यूटेट कर रहे हैं या वेदर दे आर गोइंग टू बी मोर लीथल देन बिफोर? क्या कोई तरीका कार है जिससे हमें पता लग सके, या जस्ट मतलब विजिबली क्लिनिकली हम देखते हैं डेथ रेशियो ज्यादा हो रही है तो ये लीथल हो रहा है?
बेसिकली हमारा तो हिट एंड ट्रायल का ही मेथड है, दैट्स व्हाई वी आर सो कंफ्यूज. अदरवाइज रेस्ट ऑफ द वर्ल्ड इज नॉट ओनली मैपिंग द जेनेटिक कोड, दे आर मैपिंग बेसिकली द प्रोटीन. जो स्ट्रक्चरल बायोलॉजी और मॉलिक्यूलर बायोलॉजी पढ़ने की जरूरत थी वो इसीलिए थी कि जब तक हमें ये नहीं पता होगा कि कौन सी जीन जो है किस एंजाइम को कोड करती है. जैसे मैं आपको बताता हूँ कि वायरस के अंदर एक एक्सोन्यूक्लियेज है. एक्सोन्यूक्लियोज की क्या इंपॉर्टेंस है? उसकी इंपॉर्टेंस थी कि जो राइबाविरिन थी वो राइबाविरिन को वैसे डिसफंक्शनल कर देता था. तो अब जैसे देखें रेमडेसिविर है, रेमडेसिविर एक्ट कर रही है, हम कह रहे हैं वो भी जीन्स पे ही एक्ट कर रही है ना. जीन्स जो इसका जेनेटिक जीन्स के जरिए ही प्रोटीन्स बन रही है ना, एंजाइम बन रहे हैं. जैसे आरएनए डिपेंडेंट आरएनए पॉलीमरेज है. तो अगर हम समथिंग जेनेटिकली कुछ कर सकते हो तो दैट्स मोर स्ट्रॉन्गर देन एनी ड्रग जो हम ट्रीटमेंट के लिए यूज कर रहे हो. देखें फॉर एग्जांपल अगर हम रिकम्बिनेंट, मैं आपको मिसाल देता हूँ, इफ यू कैन क्रिएट अ ह्यूमन रिकम्बिनेंट ह्यूमन एस रिसेप्टर और हम मोनोक्लोनल एंटीबॉडी की फॉर्म में इंजेक्ट कर देते हैं तो वायरस वैसे ही सारे का सारा आप कहीं के कोटेड हो जाएगा और वायरस को जगह ही नहीं मिलेंगी, ह्यूमन सेल ही नहीं मिलेंगे इन्फेक्ट करने के लिए. देयर आर सो मेनी वेज ऑफ वर्किंग. देखें इसमें इसलिए जेनेटिक्स और ये सारी चीजें हमें थोड़ी थोड़ी पढ़नी चाहिए. अच्छा, मैं एक और बड़े मजेदार चीज मैंने पढ़ी है, आई एड इट. हमारा ख्याल है कि एक स्ट्रेन से जब हम इन्फेक्ट हो जाते हैं, तो मैं तो यही समझता था कि वंस आई एम इन्फेक्टेड और समबडी इज इन्फेक्टेड विद वन स्ट्रेन ऑफ वायरस ही कैन नॉट बी री इन्फेक्टेड. ऐसा नहीं है. वायरसेस जो हैं रिकम्बिनेट करते हैं. सो वन पर्सन कुड बी इन्फेक्टेड विद टू स्ट्रेन, गॉड नोज हाउ मेनी स्ट्रेन. अगर उनके फिनोमिनोलॉजिकल पैटर्न कैरेक्टरिस्टिक वायरसेस के चेंज हो चुके हैं तो वो जरूर हो. इसीलिए जब हमारे अंदर ही एक वायरस से मिलियन ऑफ वायरसेस बनते हैं तो दे ऑल हैव सर्टेन काइंड ऑफ म्यूटेशन. जरूर उनके अंदर, हम जो म्यूटेशंस की बात कर रहे हैं ना, ये पॉइंट म्यूटेशंस हैं. ठीक है, क्योंकि हमने कहा है कि जो एनालिसिस हो रहा है वो शॉर्ट रीडिंग फ्रेम का एनालिसिस हो रहा है. पूरे पूरे बड़े जीन का, वाइड जीनोम के एनालिसिस बहुत कम हो रहे हैं. और देखें उस म्यूटेशन की इंपॉर्टेंस है जिसका कोई कॉन्सिक्वेंस भी हो, जिसका कोई नतीजा हो. बहुत सारे रीजन्स जो जीनोम के हैं वो नॉन ट्रांसक्राइबेबल है. हमें नहीं पता कि वो डायरेक्टली किसी प्रोटीन में तो आप कहें कि ट्रांसलेट नहीं होते, बट दे डू हैव अदर फंक्शन. तो इसीलिए सबसे पहले वायरस का पढ़ा जाना बहुत जरूरी है.
सही. इसमें सर एक और क्वेश्चन भी था, आपने ना एक स्लाइड दिखाई जिसमें आपने 3 रिसेप्टर्स दिखाए थे, डिफरेंट रिसेप्टर, एस2 रिसेप्टर दिखाया था और एक मोनोक्लोनल एंटीबॉडी के डोमेन का रिसेप्टर दिखाया था, एक तीसरा रिसेप्टर दिखाया था. इसमें एक तो हम आप बात करते हैं कि इंटरल्यूकिन 6 जो हम मतलब यानी कि ये टॉक्सिडोजोमैब मतलब यानी कि देते हैं इसको इनहिबिट करने के लिए ताकि वायरस इसके ऊपर अटैच ही ना हो सके. दूसरा जो जो एस इनहिबिटर एस रिसेप्टर है सर, तो थ्योरेटिकली ऐसा नहीं है कि जो बंदे ऑलरेडी एस इनहिबिटर्स ले रहे हैं या ऐसी कोई ड्रग ले रहे हैं उनके अंदर इसकी इन्फेक्टिविटी कम होनी चाहिए एज कम्पेयर टू अदर पर्सन, कि ऑलरेडी वो रिसेप्टर उन्होंने ब्लॉक किए हुए हम जो ड्रग खा रहे हैं?
नहीं, बेसिकली रिसेप्टर ब्लॉकेज से नहीं है. जो एस वाली थ्योरी है कि हाउ एस रिसेप्टर, पहले कहा गया था कि टेकिंग एन एस रिसेप्टर और ए आर बी इज डेट्रीमेंटल, मतलब इट विल कॉज मोर डैमेज टू पीपल विद इफ दे आर इन्फेक्टेड विद कोरोना वायरस. एंड उसके बाद से डब्ल्यू एच और सारों ने दे रिपील्ड, उन्होंने कहा नहीं ऐसा नहीं है. एसीसी की भी, अमेरिकन कॉलेज ऑफ कार्डियोलॉजी की भी आ गई थी फॉरन ही उसके ऊपर कि नहीं, ये बेसिकली इसका प्रोटेक्टिव इफेक्ट है. तो प्रोटेक्टिव इफेक्ट रिसेप्टर से देखें कि आगे क्या होता है. रिसेप्टर से आगे जाएं तो एंजियोटेंसिन काइंड ऑफ वी सी इन दो टाइप्स हमें नजर आती हैं. एक वेजोडाइलेटर एंजियोटेंसिन नजर आते हैं और एक वेजोकंस्ट्रिक्टर एंजियोटेंसिन नजर आते हैं. जो वेजोडाइलेटर एंजियोटेंसिन हैं, जो एंजियोटेंसिन 1/7 है, एंजियोटेंसिन 1/7 इज द मेन थिंग दैट इज प्रोटेक्टिव. इवन ओल्ड एज में भी देखा गया है कि लोगों के एंजियोटेंसिन 1/7 के लेवल्स कम थे और वेरियस डिजीजेज में सीओपीडी वालों के अंदर या कई के अंदर उनके वो लेवल्स कम थे. और ACE2 की बाइंडिंग से बेसिकली होता यही है वायरस की, ये सिर्फ रिसेप्टर ब्लॉकेज नहीं है, जो वेजो कंस्ट्रिक्टर साइड है सारी की सारी वो आप कहीं एम्पलीफाइड हो जाती है। वेजो एंजियोटेंसिनोजन, एंजियोटेंसिन 2 ज्यादा बनता है ना, एंजियोटेंसिन 2 बनेगा तो वो वेजो कंस्ट्रिक्टर सारी कैस्केड ऑफ वेजो कंस्ट्रिक्टर डैमेज जो है उससे स्टार्ट हो जाता है।
ठीक हो।
हाँ सर, आप कह रहे थे ना कि डिफरेंट एस्पेक्ट्स हर कोई बता सकता है, तो मैं थोड़ा शेयर करूंगा। अभी जैसे मैं हूँ सेंट्रल पार्क हॉस्पिटल में, जहाँ पे ना आईसीयू हम मैनेज कर रहे हैं, कोविड आईसीयू।
जी सर।
अच्छा, आईसीयू में 5 पेशेंट हैं इस वक्त। तो अभी मुझे स्टाफ ने फोन किया था, हम चूंकि ऑफिस वहाँ होती हैं। अब वो स्टाफ ने फोन किया कि जी एक पेशेंट है जिसको ना नेजल कैनुला के थ्रू हाई फ्लो ऑक्सीजन जा रही है। उसने ना जनाब हाई फ्लो नेजल कैनुला हटा दिया, कहता है जी मैंने नहीं लगाना। अब जब उसने उतार दिया तो उसकी सर्कुलेशन ना 40 हो गई। तो अब समझ नहीं आ रही उसको क्या करें। तो मतलब इस तरह की सिचुएशन हम लोग डील कर रहे हैं। मैंने ना फौरन अपने वो कोविड वाले ग्रुप पे मैसेज किया कि जी ये देखिए अब इस सिचुएशन में मैं क्या करूँ।
जी बिल्कुल।
इसमें तो एक्सपीरियंस ही काम आएगा अकमल भाई आपका। इसमें
क्या
स्टडीज
आएगा?
काम नहीं है, इसमें आपका एक्सपीरियंस ही काम आ सकता है सिर्फ,
और साथ में आपकी काउंसलिंग।
जैसे आप काउंसल कर
हाँ,
पाएंगे।
बस काउंसलिंग ही है।
बिल्कुल जी।
जी जी।
तो फिर स्टाफ ने ही थोड़ी देर में उसको काउंसल किया, कोई चाय की ऑफर की, बड़े उससे ही ना उसने
चाय के
मान
साथ
लिया।
बिस्कुट भी करते तो फिर तो पक्का हो जाना था।
वो जनाब थोड़ी देर बाद मैंने रात दोबारा किया, तो वो जनाब उसकी 93 सैचुरेशन है और उसने लगवा दिया। चाय की चाय
वाह।
पहले ले आओ। तो ये
माशाल्लाह,
सूरत हाल है।
देखिए फायदा हो गया। ऐसे ही है।
ये
तो
एक्सपीरियंस का फायदा हुआ ना
मैंने तो बड़े
सबका।
कड़वे मैसेज ना मैंने ना ग्रुप पे बड़े कड़वे मैसेज कर दिए। मैंने कहा जी ये देखिए हम तो टार्जन की औलाद थोड़ी है, इसको हम क्या करें। बताएं, मैंने कहा मशवरा दें। बड़े दिन वाले ना बड़ी बातें कर रहे थे क्योंकि वो बार बार जाके खुद प्रोनिंग कराए मरीजों की। वो मैं कि भाई ये नर्सिंग केयर है, ये हमने नहीं करना। और
सर एक्चुअली
मैंने
ना
कहा एक
लोगों में जो फियर
एक
है ना,
लाइफ
एक
तो आप कर नहीं रहे।
फियर बहुत ज्यादा है
हाँ
लोगों में इसका। लोग हॉस्पिटल में जा
हाँ जी
नहीं
हाँ जी।
रहे, प्राइवेट लेवल में बहुत पेशेंट है, ओपीडी में भी आते हैं, बट वो हॉस्पिटल से डरते हैं, कहते हैं जी डॉक्टर साहब टीका लगाते हैं मार देते हैं।
जी ऐसे ही है सर।
चलें सर, थैंक यू वेरी मच, बहुत
ओके
अच्छा
जी,
हो गया।
नाइस मीटिंग
सर
है,
माय
बहुत अच्छा
प्लेजर,
रहा, बिल्कुल।
माय प्लेजर। इंशाल्लाह
थैंक यू।
विल कीप ऑन डूइंग। अब नेक्स्ट
रिकॉर्डिंग
आप
शेयर
में से
कर
कोई।
दीजिएगा, मैं फिर आगे ग्रुप।
बिल्कुल।
जी
मैंने तो
इंशाल्लाह।
कुछ स्क्रीनशॉट भी ले लिए हैं उनके, बड़े अच्छे अच्छे वो आपने बनाए हुए।
यू आर मोर देन
वो
वेलकम।
आराम से बंदा पढ़ भी लेता है बाद में इजीली।
श्योर, एनी टाइम आई अपलोड इट। मैं इसको वैसे भी
थैंक यू सर।
अगर ना भी हो अलग से भी मैं अपने
थैंक यू
यूट्यूब
वेरी मच।
चैनल पे भी अपलोड
जी
कर।
जी। जी जी,
थैंक
ओके
यू
सर,
वेरी
टेक
मच।
केयर,
ठीक
बहुत
हो
अच्छा
गया,
रहा।
थैंक यू सर।
ओके, अल्लाह हाफिज़।
ओके, अल्लाह हाफिज़।
अल्लाह हाफिज़।
अल्लाह।